Okay, good evening, everyone. So I'd like to thank the organizing committee for the possibility to present our work. And can you see my presentation now? Today I'd like to tell you about 3DPredictor algorithm. The main idea. First, we want to predict spatial chromatin contacts in normal cells. And when we want to predict the changes in these contacts after chromosomal rearrangement. And also in future, we want to predict some changes in gene expression. And also it's interesting to check this model experimentally, for example, on mouse model. Here is the illustration of model creating. So what kind of information can you use for prediction of 3D chromatin structure? It considers that some epigenetics data correlates with 3D genome structure. So initially, we used all available data from ENCODE as RNA-seq data, ChIP-seq data for different proteins, and so on for prediction. And to found these complex patterns, we used gradient boosting regression and machine learning algorithm. So then we parameterized this epigenetics data and split our data to non-overlapping train and validation data sets. So firstly, we got predicted high-C map like here on this picture, and as you can see, this high-C map was predicted with low efficiency. Actually, as you can see, we just predicted the decay law of contact probabilities with distance. So after that, we tried to modify some algorithm parameters, the sample, the size of training data sets, and so on. And finally, we got the high-C map, like here. Here's the prediction for mouse hepatocytes. And as you can see, the algorithm captured the main text structure, and sometimes it even predicted loops. But this picture is quite rough assessment of algorithm efficiency. So further, we used more strict metrics, and the main metric was stratum adjusting correlation coefficient, which was proposed by Jan and co-authors in 2017 for comparison of distant high-C map. And firstly, we compared differences between high-C maps of replicates. Then we compared high-C maps of distant cell types and compared the differences between predicted data and real high-C data. And as you can see, the differences between predicted and experimental high-C data closer to the differences between replicates than differences between distinct cell types. So in general, this algorithm really captured the main 3D genome structures, but this prediction is not perfect. So the algorithm has its own limitations. For example, I think everyone here knows that the main features of high-C maps are tests and loops. So how well 3DPredictor captures loops? And here's the predicted high-C map. And as you can see, sometimes this algorithm really captures loops, like here on this case, but sometimes it fails. So, but in general, yes, this algorithm predicts the main 3D genome structures. So after that, we try to investigate the black box of gradient boosting. So we extracted the feature importances, in other words, the contribution score for every predictor, and shows the most important predictors and the most sufficient predictors as RNA-seq data, CTCF, chip-seq data, orientation of CTCF binding sites, and genomic distance between loci. So this subset of predictors is sufficient to predict Hi-C-map like this with high accuracy. And also different cell types has different available proteins, but these predictors are almost always available, for example, in ENCODE database. So we used mainly this subset of predictors. But when we analyzed feature importances for different cells, for different cell types, we found some unexpected results. For example, when we extracted feature importances for K562 cell line, we found that the major predictor is the protein ARID3A, and it's strange. So still we don't understand, is it an artifact, or maybe this protein really has biological meaning. So just interesting fact that requires further investigation. But now let's return to our prediction, and also we tested how well we predicted cell type specific chromatin organization. And here you can see experimental Hi-C data for neuroprogenital cells and mouse hepatocytes, which differs by this TET boundary. And below our predictions, indeed, the algorithm captured this difference. It's predicted the boundary, and there is no TET boundary for mouse hepatocytes. And in general, we have an algorithm which can predict 3D genome organization for different genome loci. So we have an online web tool for prediction. You need RNA-seq data and CTCF chip-seq data for prediction. And then you can use this web tool and try to predict locus you're interested in. So here in this QR code, the link to this web tool. So this you can try to predict Hi-C map for locus you're interested in. And I started my talk with the idea to predict changes in 3D genome contacts after chromosomal rearrangement. So here's the experimental data for mast cells. And please pay attention to this TET boundary. Here's this zoomed region. So there are 4 CTCF peaks located in this border. So what happened if we delete this region? So we try to predict these changes using the algorithm. So here the prediction for wild type, and as you can see, the algorithm captured this TET boundary. And here's the prediction for deleted region. So the prediction of consequences of rearrangement. And the algorithm predicted that these TETs are merged. So my colleagues from the lab created transgenic mice with these deletions, with this deletion, and also we thought that these genes KIT and KDR also change the expression. but there is no changes in gene expression. But also we did high-C experiment, and here you can see the real data for mutated cells. And as was predicted, really these stats are fused here. But it's only the one case of chromosomal rearrangement. So we decided to extend the data set of rearrangements, and we analyzed the literature and collected 77 rearrangements with available high-C or capture high-C data for mutated cells for 8 cell types from 11 articles. There are the majority of rearrangements are deletions, but there are also some duplications and inversions. And the main idea to compare the ability of algorithm to predict consequences of different chromosomal rearrangement. And also we want to compare the prediction efficiency of all existing algorithms. If you have an algorithm which can predict consequences of chromosomal rearrangements in 3D genome contacts, we offer you to join to this project. Here in this QR code, the link to the dataset, so you can browse this dataset. And also, if you have some experimental data for rearrangements, which leads to changes in 3D genome contacts, and you have capture high-C data for these cells, also we offer you to join to this project and submit your data to the existing dataset. So that's all about 3DPredictor, and I acknowledge all colleagues from our development lab from Institute of Cytology and Genetics, and also thanks for organizing committee. Now I'm ready to answer your question.
Thank you very much. I think Ivo had a question.
Yes. So I was wondering which amount of data do you need, and I'm more interested in whether you can hopefully think that it can be applied to bacteria.
No, we can't apply this algorithm for bacteria because when we used machine learning algorithms, we think that this algorithm captured the main complex patterns. And we used, for example, CTCF data, and we know that loop extrusion works for mammalian genomes. So this can work only for mammalian genomes. But if you trained the model again using data for bacterial, you also can capture some complex patterns and try to predict something for bacterial too.
Yes, but it's not clear whether you have enough information or enough training data for bacteria. I mean, for the case of human genome, I guess, yeah, you can train on different chromosomes and apply your tool on a different chromosome. But in bacteria, I mean, or you may expect that your knowledge from one bacteria can be transposed to another bacterium. But I mean, that's open question, I guess.
Yeah, maybe we haven't sufficient data for prediction. I don't know what data we have for bacteria, for example. So we can try to predict that.
I will.
So there are many questions. Unfortunately, we won't be able to tackle all of them. One question is about, do you think it's possible to predict the chromosome organization of patients using mutations or copy number variation? So using high-C data from control.
As I understand the question about can we predict some mutations for patients? Yes.
Yeah, so can you predict high-C maps for patients that have cancer using information about mutations, copy number variation, and a high-C map from a non-cancerous tissue?
Exactly, we need not a high-C map, we need CTCF, chip-seq data, and RNA-seq data for these patients for prediction. And yes, in this case, we can try to predict the high-C map, the changes in this high-C map for these patients. If we have CTCF, chip-seq data, and RNA-seq data.
There's another question about, can you give me a bit more detail on how the algorithm works and, for example, how the training data is defined, which you've hinted already in some of your answers to questions?
As I understand the question about the training data set, yes, how we train the model. I don't know, it's really a complex question because it's gradient boosting, and actually the main part of an algorithm is preparing of data for training. Also the main part is parameterization of this data. So for example, we use CTCF chip-seq data. So what we do with this data, we use, for example, signal value information for the window between two contacts. It is one predictor. Also, for one protein, there are some more predictors. For example, we used information about 3 nearest CTCF peaks, of one contact, and so on. So there are, I don't know, I can't tell you everything about data preparation, but you can read our paper.
And so maybe one last question. What were the most important hyperparameters to tune in order to improve the prediction from the initial results you showed at the beginning of your talk to the results you showed at the end?
Yeah, okay, thank you. I think that it was data parameterization. So when we change, for example, the size of training data, we had small we had small so if answer to the question, the main improvement was the data parameterization, not the size of training, not the algorithm parameters. It was a data parameterization.